Today we will discuss the topic life cycle of Chara. Systematic position of Chara. It belongs to division Thallophyta, subdivision Algae, class Charophyceae, order Carylis, family Cariaceae, and the name of the genus is Chara. Chara, it is also known as stonewort. What is the habitat or occurrence of Chara? Chara is a submerged aquatic alga of freshwater ponds, tanks, lakes, or slow-running streams. Plant body is attached to the muddy or sandy bottoms with the help of rhizoids. Some of the species, for example, Chara baltica, occurs in brackish water. Brackish water means it is found in marine water, but the salt concentration is more. Then one of the species, Chara fragilis, occurs in hot springs. Some of the species of Chara get encrusted with calcium carbonate so that the species become hard and brittle. Therefore, it is called as the stoneworts. Then let us see here the thallus structure of Chara. Plant body of the Chara is macroscopic, well-branched and erect. What is thallus? Thallus means the plant body is not differentiated into root, stem and leaves. Therefore, it is called as the thallus. So, in Chara, thallus is macroscopic. Macroscopic means it is easily seen by naked eyes, then well-branched and erect. The height of the plant body is 20 to 30 centimeter in length. The plant thallus is differentiated into many rhizoids and main axis. These are the rhizoids and this is the main axis. Rhizoids are thread-like, uniseriate, multicellular, branched and white structures. Rhizoids keep the plants attached in the mud or sand. Nodes and internodes are absent in rhizoids. Then the main axis. Main axis differentiated into nodes and internodes. Main axis is erect and well-branched. Internode is long, cylindrical, undivided, single cell. Node of the main axis is short and consists of a pair of central cells in one tier. From the node of the main axis develop 4 types of appendages, and these 4 types of appendages are branchlets, branches of unlimited growth, stipulodes, and cortex. This is the main axis, and this main axis is erect and well-branched, and consisting of 4 types of appendages, and these 4 types of appendages are branchlets. These branchlets, they are also called as the branches of limited growth, and they are also known as primary laterals, and they are of short size. Therefore, they are also called as branches of limited growth. Then another is the branches of unlimited growth. This is the branch of unlimited growth. It is continuous in growth. Then there are stipulates are also present and the cortex is also present. Stipulates, cortex, these are the long internode cell of the main axis remain covered by a cortex. Then let us see here reproduction in Chara. Chara reproduces either vegetatively or sexually. Means in Chara there are 2 types of reproduction: vegetative reproduction and sexual reproduction. First, let us understand here vegetative reproduction in Chara. Vegetative reproduction takes place by these 4 methods, and these 4 methods of vegetative reproduction are bulbils, amorphous bulbil, secondary protonema, and amylum stars. What are bulbils? Bulbils are rounded bodies found either at the root nodes or stem nodes. These are the bulbils. They are present, found either at the stem nodes or the root nodes. On being detached from the parent plant, these bulbils germinate and gives rise to a new plant. Then another method, that is the amorphous bulbil. In some of the species of Chara, numerous small cells aggregate on stem, root or root nodes and form many lateral outgrowths called amorphous bulbils. These are the amorphous bulbils, and these amorphous bulbils get detached from the parent plant and they give rise to new plants. Then third method, secondary protonema, either from the rhizodal node of the primary protonema or from the basal cell of the primary rhizoid, may develop some tubular bodies called secondary protonema. Then the fourth method of vegetative reproduction, amylum starch. In few species, amylum starch containing cells aggregate on the lower nodes of the main axis, and these bodies appear star-shaped. These contain amylum starch. So, on being detached from the parent plant, these amylum starch develop into new plants. These are the amylum stars. These amylum stars, they are star-shaped bodies, and these star-shaped bodies contain amylum starch. And when they get detached from this plant body, amylum stars, they give rise to a new plant. These are the four methods of vegetative reproduction. What are the four methods? Bulbils, then amorphous bulbils, secondary protonema, and amylum stars. Let us see here sexual reproduction in Chara. Chara reproduces by an advanced and specialized type of oogamous sexual reproduction. Here, what is oogamous type of sexual reproduction? Oogamous type of sexual reproduction means here the female gamete is larger in size and non-motile, and the male gamete is smaller in size and motile. And when the fusion takes place between these two gametes, this is called as the oogamous type of sexual reproduction. So, here in Chara, oogamous type of sexual reproduction. In Chara, the sex organs are well developed and visible to the naked eye. These are the sex organs, and they are well developed and visible to the naked eye. The male reproductive bodies are spherical, bright yellow or red structures called antheridia or globule. In Chara, the male reproductive organ is known as the antheridium or globule, and the female reproductive bodies are oval green structures called oogonium, or it is called nucleus. Here, one thing here, the male reproductive organ is known as globule, and the female reproductive organ is known as the nucleus. Most of the Chara species are homothallic and few are heterothallic. What is homothallic species and what is heterothallic species? Homo means same, thallic means thallus, hetero means different, thallic means thallus. Homothallic means both the sex organs, that is antheridium and oogonium, occurs on the same thallus. And heterothallic means the reproductive organ, that is the male reproductive organ, is present on one thallus, and the female reproductive organ, that is the nucleus, it is found on different thalli. That is called as the heterothallic. Here, this is the homothallic species. This is branch of the Chara, and here both the reproductive organs, they are present on the same thallus, antheridium and the oogonium. So, in Chara, most of the species, they are homothallic, and a few species are heterothallic. Then the position of the sex organ is here, the nucleus is present above the globule. This is globule and this is nucleus, and this globule is present above the, this nucleus is present above the globule. Then let us see here the structure of male reproductive organ. In Chara, male reproductive organ is known as the globule. This is the diagram of globule. This is a globule. A mature globule or antheridium is a large, bright yellow or red spherical body. It consists of 8 closely fitted curved plate-like shield cells, 8 rod-shaped radially elongated manubrial cells, 8 centrally situated primary capitulum cells, and about 48 secondary capitulum cells. From the secondary capitulum cells develop long thread-like multicellular antheridial filaments, and antheridial filaments may be branched or unbranched, and each antheridial cell functions as a sperm mother cell. Each sperm mother cell gives rise to a single uninucleate biflagellate coiled antherozoid or spermatozoid. From these secondary capitulum cells develop antheridial filaments. These are the antheridial filaments, and each antheridial filament contains about 100 to 1000 cells. And each antheridium cell, then the nucleus of each antheridium cell metamorphoses into a single antherozoid or the spermatozoid. This is the antheridial filament, and this antheridial filament contains 100 to 1000 cells, and each antheridial cell contains single nucleus, and this single nucleus metamorphoses into a single sperm or male gamete, or it is also called as antherozoid or the spermatozoid. So in one antheridial filament there are about 100 to 1000 antherozoids. Now, how is the liberation of antherozoids? How is the liberation of antherozoids? These antherozoids are uninucleate biflagellate coiled structures. This is antherozoid or the sperm or the male gamete, and these antherozoids are coiled uninucleate biflagellate structures. When the globule is mature, the shield cells separate from each other, and the manubria, capitula, and antheridial filaments are exposed, and the antherozoids liberate by the gelatinization of wall of antheridium. Then let us see here female reproductive organ. Structure of female reproductive organ. In Chara, female reproductive organ is known as the nucleus or the oogonium. So here, see here, this structure of nucleus or oogonium. This is the structure of nucleus. The oogonium or nucleus is an oval structure attached on the node with the help of a pedicel cell. It remains surrounded by 5 long twisted tube cells, at the top of which is present a coronary cell. These are the coronary cells, and there are 5 coronary cells. This is the structure of nucleus. Nucleus is oval-shaped body attached on the node with the help of this pedicel cell. It remains surrounded by 5 long spiral twisted tube cells, and at the top of which tube cell there are 5 coronary cells. The oogonial cell contains a single large egg or ovum. This oogonium contains a single large egg cell or the ovum. This is the egg cell or ovum. And the mature egg contains single nucleus and a receptive spot on the top. Now, after the formation of both the gametes, that is the male gamete and the female gamete, the process of fertilization takes place. How is the process of fertilization? In Chara, fertilization takes place with the help of water. So at the time of fertilization, the tube cells slightly separate from one another near the coronary cells, and this results into the formation of 5 narrow slits. Then the apertures are filled with colorless viscous liquid to which the swimming antherozoids attached or adheres, and later one of the antherozoids penetrates the ovum near the receptive spot and fuses with the egg nucleus to form diploid oospore. What happens here after at the time of fertilization? The tube cells slightly separate from one another near the coronary cells. This results into the formation of 5 narrow slits. Then the apertures are filled with colorless viscous liquid to which the swimming antherozoids adheres, and then one antherozoid penetrates into the ovum near the receptive spot, and one of the male gamete fuses with the egg nucleus to form diploid oospore. Then after the fertilization, zygote is developed, and this zygote secretes a thick wall around itself, and that structure is called as the oospore. It's the oospore. It's oospore. Oospore is a hard nut-like body of various colors. It is a spherical to narrowly ellipsoid, and it remains surrounded by 4-layered wall. The 2 outer membranes are colored and 2 inner ones are colorless. Here, oospore is a hard nut-like body of various colors, and it consists of 4 membranes. The 2 outer membranes are colored and 2 inner ones are colorless. Now here, the nucleus of the oospore travels towards upper side, and the oogonial wall ruptures, and on rupturing the oogonial wall, the oospore comes out and settles at the bottom of the pond. And when the oospore settles at the bottom of the pond, it germinates after a period of rest. Now, what is the process of germination? Now, how is the germination of oospore or the zygote? At the time of germination, the diploid nucleus, this is the oospore. This is the diploid oospore. The diploid nucleus migrates towards the upper side and divides. Here, this oospore nucleus divides meiotically and to form 4 haploid nuclei. These are the 4 haploid nuclei. Now, here, what happens here? The oospore gets divided into 2 unequal cells by a transverse wall. Upper one is smaller cell and the lower one is the larger cell. Here, the upper smaller cell contains single nucleus, and the lower larger cell contains remaining 3 nuclei. Now here, the 3 nuclei containing lower larger cell degenerate, and only one nucleus remains functional of the upper cell. Now here, the nucleus of the upper cell project out by rupturing the oospore wall and divides by an oblique longitudinal division to form protonymal initial and rhizoidal initial, and both the initials grow in opposite directions. Here, this uninucleate upper nucleus projects out by an oblique septa and grows into 2 opposite directions. One forms the rhizoidal initial and other forms the protonymal initial. Later, this protonymal initial, the rhizoidal initial develops into colorless rhizoid, and protonema initial develops into an erect primary protonema. That means again a new plant body develops, and then this primary protonema gets differentiated into nodes and internodes. Now here the life cycle of the Chara is complete. Thank you.